Dear students, in this video we are going to study the one of the important trematode, that is the Fasciola hepatica, and it's commonly known as the liver fluke. So liver flukes are typical Digean trematode and are commonly called flatworms or flukes on account of their flat leaf-like structure. Fasciola hepatica is the common liver fluke of sheep. It is first trematode whose life history was described by Thomas in 1883. It is of much importance as it causes fascioliasis, a disease that causes damage to liver tissues and bile ducts of sheep. The parasite has a widespread distribution throughout many sheep and cattle raising countries of the world. Numerous human infections have been reported from Latin America, France, England, Germany, Poland, China, and some parts of Africa, and in some parts of the southern continental United States. The adult fluke is flattened and leaf-like, with an anterior conical projection and a broadly rounded posterior end. The adults, which measures up to 3 cm in length and 1.3 cm in width, and produce the operculated eggs. External morphology: the anterior end of body is distinguished into triangular oral cone or head lobe. Head lobe at its tips bears a somewhat triangular aperture called as mouth. There are two muscular suckers or olar suckers and a large ventral sucker or acetabulum, which is situated mid-ventrally. In front of the ventral sucker is common genital aperture or gonopore, and at the posterior end of body called as the excretory pore, temporary opening of Laurus canal. The sheep liver fluke is an endoparasite which completes in its life history in two hosts. The adult flukes are typical parasites of vertebrate animals, but one stage of their life history is invariably spent in an invertebrate host or mollusks. So in its life history, they are having the two hosts. They have the digenetic life history. Sometimes the adult fluke invade other vertebrates also, such as goat, horse, dog, rabbit, monkey, and even the man. They cause serious loss of human life and domestic animals. Though Fasciola hepatica is hermaphrodite, even then cross-fertilization is of common occurrence. The eggs are fertilized in the oviduct. The fertilized eggs receive yolk cells from vitelline glands and they get enclosed in the chitinous shells. The completed eggs are called capsules, which are large in size and they pass into the uterus. The capsules come out of the gonopore into the bile duct of sheep. They reach the intestine and are passed out with the feces. So this is the structure showing the male and female reproductive structure of Fasciola hepatica, as it is a hermaphrodite. So this is the life history of Fasciola hepatica. You can see here that there are the two hosts. First host here, you can see the life cycle in the sheep, and here you can see the life cycle in the snail. So as a encysted Cercaria larva, it just taken up by the sheep and it will change into the adult fasciola, then giving rise to the a capsule formation and some larva formation which is a Miracidium. Then Miracidium will change into Sporocyst. Sporocyst will give rise to Redia, and Redia will give rise to Cercaria, and Cercaria will change into the Metacercaria. So the stages from Metacercaria till the adult one, these are passed in the sheep, and stages from Miracidium till the Cercaria larvae, these are passed in the snail. So when the conditions turn favorable, uh, this is the Miracidium larva. So you can see the different forms as it is showing the polyembryony. So first larva form here is going to become the Miracidium. You can see the structure of Miracidium here, showing the presence of apical papilla, apical gland, presence of cilia all over, presence of brain, eye spot, excretory pore, penetration gland, epidermal plates, flame cells, germ balls, etc. So when the conditions turn favorable, the encapsulated embryos get differentiated into Miracidium larva. So this is how it changed into the Miracidium larva. So it is the first larva stage in the life cycle of liver fluke, and these larvae are hatched out with the help of hatching enzyme. So this is the structure of Miracidium larva. So Miracidium larva is oval, small, elongated and richly ciliated. Anterior end produces the non-ciliated apical papillae, and this larva is multicellular and body is covered with ciliated epidermal plates, which are total 21 in number and arranged in 5 rows or tiers. The number of plates in each tier is fixed. So as if you go into the details of these plates, so Miracidium larva is minute oval elongated free swimming stage. It is covered with 18 to 21 flat ciliated epidermal cells lying in 5 rings. First ring is made of 6 plates, 2 dorsal, 2 lateral, 2 ventral. Second ring has 6 plates, 3 dorsal, 3 ventral. Third ring has 3 plates, 1 dorsal, 1 2 and 2 ventrolateral. Fourth ring has 4 plates, 2 right and 2 left. And fifth ring has 2 plates, 1 left and 1 right. You can see through the diagram. You just go to the diagram. So next is the Sporocyst larva. So this is the structure of Sporocyst larva. It is slightly elongated structure which retains the body layers of previous larva stage except the ciliated epidermis. There are no cilia here, and this is replaced by the cuticle, and the glands, brain, eye spots, apical papilla which are present in the Miracidium, they now get degenerated in this stage. You can see here the patches of cuticle, then muscle layer. There are patches of flame cells, proton leading into the opening into the protonephridial openings. There are patches of germ cells, and there are patches of just the developing redia. So this is how the Miracidium larva will give rise to the next stage, that is the Sporocyst larva. Next larva is the Redia larva. So in the developing Sporocyst larva, there are the development of Redia which emerge from the Sporocyst larva. So it is somehow rectangular, and germ bars are present uh, which were present in Sporocyst. They develop into the Redia larva. So Redia larva come out of the Sporocyst by rupturing its body wall, and these are elongated and cylindrical structure. You can see the patches of mouth, pharynx, pharyngeal glands. With the collar, there is patches of gut, and there is patches of birth pore, and there are the developing Cercaria inside the Redia larva. And there are the patches of cuticle, epithelial cells, and you can see at the posterior end there are the patches of lappets, and germ cells inside the radial larva are also present. So they both have the mouth, they have mouth at anterior end and a ring of collar cell with the birth pore present at the anterior end. So body wall having the cuticle, longitudinal muscles and subepithelium. Mouth, as already explained, leads into pharynx, and that leads into intestine. Numerous pharyngeal glands are also present, protonephridia are also present, and body of this larva is packed with the germ balls and mesenchyme cells. Now the next stage is the Cercaria larva. So each of the radial larvae of the second generation give rise to 20 Cercaria larvae. They leave the body of Redia from the birth pore present at the anterior end and then enter into the digestive glands of the snail. You can see here the patches of Cercaria larva. It is somewhat showing the oval structure, having the body showing the oval structure, and there is the presence of tail. And in the body you can see the mouth, oral sucker, musculopharynx, gut with the bifid intestine, and presence of acetabulum, presence of germ cells, presence of flame cells, etc. So Cercaria larva is oval structure with tail. Tail helps in swimming, and cuticle is also present. So body wall consisting of cystogenous gland cells which secrete cyst for the next larval stage. So next larval stage is metacercaria larva. So it is somewhat circular, spherical, and as the cyst wall, thick, hard, whitish cyst wall is formed around the cercaria larva, which is formed by cystogenous glands. So it forms the metacercaria. So this encysted larva must be ingested by the primary host again. So if unencysted larva is ingested by the sheep, they would not be digested by the acidic action of sheep's gastric juice. So this is the encysted form of Cercaria larva. So life history of FASCIOLA hepatica affords a good example of alternation of generation, as fluke is a sexual generation and it alternates not with an asexual generation but with parthenogenetic generations of Sporocyst and Redia. Such an alternation of sexual generation with a series of parthenogenetic generation is called heterogamy. So asexual multiplication of various larvae is called polyembryony. So you can attempt this question, the life cycle, or showing the giving the different larva forms of FASCIOLA, as if asked about the polyembryony also, or if there is a question of heterogamy. So heterogamy as well as a polyembryony as well as the different forms, larva forms or life cycle of the FASCIOLA hepatica. Pathogenicity. So disease caused by the infection of FASCIOLA hepatica is known as fascioliasis. It mainly affects the liver, but it also causes hepatitis and inflammation in the bile duct. Due to this, the bile ducts become thickened, which is followed by calcification and finally resulting into the formation of gallstones. During heavy infection, the liver fluke seriously affects the liver of sheep and its functions are upset. The sheep gets a disease called river rot. Due to irritation by means of their cuticular spines, muscles become weak and functions of the liver are upset. So these are the diagrams or pics showing the bile duct inflammation, gallstone, liver rot in sheep and hepatitis. So its infection to the secondary host snail causes complete destruction of the digestive glands or the site of infection, finally causing the death of the snail. Sometimes infected snails attain gigantic size. Treatment and control measures. Antihelminthic drugs like hexachloroethane, carbon tetrachloride, tetrachloroethane, etc. can be used. The For prophylaxis, the killing of secondary host or snail can be done by introducing ducks to the pastures, which will rapidly killing of having heavily infected sheep, destroying the eggs and manure of infected sheep, and spreading of its infection can also be prevented by treating the infected sheep effectively with hexachloroethane, which can destroy most of the fluids. So these are the different larva forms of the FASCIOLA in the life history of FASCIOLA. You can see here first this one is the Miracidium, which is also showing the plates. This is the Sporocyst, then Redia larva, then Cercaria larva and then Metacercaria larva. Thanks.